A very good evening to you all, and welcome to yet another session of Educare Analytics as powered by Safe Publications. In this session, we are going to digest the relationship between cell division and cancer. The contribution of mutation in cancer development will also be discussed in the same vein. What is the relationship between cell division and cancer? Cancer is a group of disorders that result from uncontrolled mitotic cell division. Such a cell division forms a cell mass called a tumor. How? Cell division is controlled by specific genes called proto-oncogenes and tumor suppressor genes. So it stops when enough number of cells required to perform a specific function has been formed. However, when such proto-oncogenes undergo mutation, they form active oncogenes, the genes which code for proteins like cyclin that promote cell growth, division, and survival. Therefore, cell division runs out of control, whereby cells continue to divide uncontrollably, leading to development of a group of abnormal cells that divide more rapidly than normal surrounding cells. Such a cell mass formed is called a tumor. Why tumor cells continue to divide? This is because tumor cells don't undergo differentiation, thus not specialized to perform any function in the host tissue. They just interfere with the normal activity of the normal cells in the host tissue by restricting blood flow to them. Tumors can be benign or malignant. Benign tumors are slow-growing, inactive growths contained in fibrous capsule which don't invade other tissues. They never get spread, but larger ones exert pressure on blood vessels, thus restrict blood flow to the area, but are not cancerous. Therefore, they are inactive tumors which are relatively harmless. However, malignant tumors are faster-growing, active tumors which invade or spread to other tissues, thus are cancerous. This is because their cancerous cells are transported via bloodstream or lymph and form secondary tumors called metastasis in a variety of other body tissues by a process called metastasis. They develop their own blood vessels by angiogenesis and outcompete the surrounding tissues for nutrients and oxygen. Therefore, malignant tumors are the tumors which break out of their sites and spread via blood or lymph to initiate secondary cancers or metastasis in other tissues and organs by metastasis. Similarly, cancer can also be developed due to point mutations that result into inactivation of tumor suppressor genes or anti-oncogenes, which code for proteins like p53 that block uncontrolled or excessive cell division and growth, promote DNA repair, and trigger apoptosis, thereby preventing cancer development. Tumor suppressor genes are a group of genes that slow down or inhibit the rate of cell division in a developing tumor. Mutation inactivates these genes, which increases the chances of a significant growth of the tumor. Note: inactivation or dysregulation of tumor suppressor genes is more important in cancer development than activation of oncogenes. Likewise, epigenetic silencing via methylation of the promoter is vital in dysregulation or inactivation of anti-oncogenes and activation of oncogenes rather than point mutations like insertions and deletions. Hint: we develop millions of tumors every day. Fortunately, many tumors are detected and destroyed by the body's immune system. However, if the tumor cells are too similar to normal or ordinary body cells, the immune system can't detect them for destruction until the tumor is well established. The likelihood of cancer development increases with age, and the risk roughly doubles with each decade beyond the age of 30, with breast cancer and prostate cancer taking the lead. So everyone is a likely victim of cancer since these mutations occur by chance. Take home: cancer is due to uncontrolled mitotic cell division. How do mutations contribute to cancer? Mutations contribute to cancer development once they occur in two types of genes, that's oncogenes and tumor suppressor genes, or anti-oncogenes. Oncogenes are a group of genes which code for proteins that switch on cell division. These genes normally occur in their inactive form as proto-oncogenes, which code for proteins required for cell growth, division, and survival, as well as tissue repair. Some point mutations like insertion and deletion activate proto-oncogenes into oncogenes, leading to uncontrolled cell division and thus tumor or cancer development. Normally, some of their proteins are inactivated by those coded by tumor suppressor genes, which enables cell division to remain switched off. Tumor suppressor genes are a group of genes that slow down or inhibit the rate of cell division in a developing tumor. Mutation inactivates these genes, which increases the chances of a significant growth of the tumor. Causes of cancer. There are various factors which cause changes in genetic pathways and lead to cancer development. However, the rate of mutation of these genes important in cancer development is increased by the following key agents: ionizing radiations like ultraviolet radiations and X-rays. Sunbathing in strong sunlight exposes the skin to ultraviolet light, increasing the risk of developing skin cancers like malignant melanoma, which is often fatal. The ionizing radiations in nuclear fallout can penetrate all body tissues and cause leukemias and other carcinomas. Chemical carcinogens. There are several chemical carcinogens. Here are a few: exposure to tar in cigarette smoke, benzene and naphthalene in diesel exhaust fumes, and benzene in petrol exhaust fumes. Smoking significantly increases the risk of lung cancer due to exposure to tar. DDT and painkillers like aspirin, paracetamol, and other chemicals in food additives like preservatives and food colors etc. are carcinogens if they overaccumulate in the body. Not well. There are several chemical carcinogens and some are not yet identified. Viral infections. Some viruses inactivate anti-oncogenes or activate oncogenes, leading to cancer development, like human papillomavirus is linked to cervical cancer. Hepatitis B virus causes liver cancer, among others. Grab your copy of Ask Biology Kindle today and enjoy the reading. Thank you for learning with us and supporting safe publications. God bless you.